You're ready?
Yeah, I'm actually thinking this.
All right. Let me get the mic. Well.
Wow. Never before in my life I had such a silent. Please. All right. Good afternoon, everybody. And can you hear me? No?
Now, right? Thank you for coming, and please join me in welcoming our speaker today, Leila Hamden. Leila is an associate professor at the GCRL at the Gulf Coast Research Laboratory. Most of you know that lab. We have been working together for quite a long time now, and the GCRL is the physical lab that represents the Division of Coastal Sciences from the School of Ocean Science and Technology, newly formed School of Ocean Sciences and Technology, which basically brings together now the Division of Coastal Sciences and the Division of Marine Sciences from the University of Southern Mississippi. Leila got her PhD in Environmental Science and Policy from George Mason University. Then she was a National Research Council postdoc. She then worked as a research microbiologist in the Marine Biochemistry section at the Naval Research Laboratory in Washington, D.C. And then she went back to George Mason University as an assistant professor, and then she came to the University of Southern Mississippi. Leila's interest is in microbial ecology, molecular microbial ecology, and focusing on geomicrobiology, which is pretty cool. And lately she has been doing very interesting work looking at microbial biogeography on continental margins and the impacts of hydrocarbons and chemical dispersions on communities found on shipwrecks. So today she'll be telling me about that, and thank you for coming.
Okay, can everybody hear me? Great. Well, just thank you for that introduction, and thanks to you and to Ruth for the invitation to come and speak to you all today. I've already told them both that this visit to Dauphin Island is really long overdue. Now that I've been here, you're going to have trouble getting rid of me. It's just such a beautiful facility with such great science happening here, and I'm just excited to be here to talk to you all. I'm going to be presenting some of the work that myself and my team have been working on for the past several years on a study of the impacts of the Deepwater Horizon spill on deepwater shipwrecks in the northern Gulf of Mexico. And towards the end of this presentation, I'm going to take you in some of the directions that the work has wonderfully led us to, and get to this stowaways part of the title of this presentation. But before I get into that, I do want to acknowledge the many collaborators, especially my co-PIs on the SCHEMA project, which I'll define in just a few slides, especially Melanie DeMoore, Lisa Fitzgerald, and Chris Harrell, who were my co-PIs, and the many students, postdocs, and technicians that have contributed to this work over the years. So as the title hopefully tipped you off, I will be speaking about historic shipwrecks in this presentation, and to give you an understanding of what that term means, a historic shipwreck applies to shipwrecks that are greater than 50 years old. And when wrecks have that venerable characteristic to them, they are thus afforded some protection under the National Historic Preservation Act. And this act requires that federal agencies consider the effects that permitted activities have on cultural resources in the deep ocean, or in the coastal ocean as well. And the permitted activity that our work has been addressing for the past 5-ish or so years are the impacts of the Deepwater Horizon spill. And we thought that this was an important knowledge gap to fill, because the impacts to shipwrecks of the spill were not addressed under the NERDA process, not addressed by GOMRI or any other funding line. And we thought that this was important to look at because these shipwrecks represent our cultural, maritime, and in some cases our wartime heritage on the continental shelf spanning the last 500 years. And as our main project archaeologist on this program and our program officer, Melanie DeMoore, has said to me a number of times, these are 100% non-renewable resources. Once they're gone, they are completely gone. So it behooves us to extract whatever kind of information that we can about our history from them while they still exist. And at the bottom of this slide, you'll see 3 of the 7 shipwrecks that are included in our study. The Halo steam tanker, the German U-boat U-166, and the Anona steam yacht. And each of these are World War II era vessels. 2 of them are World War II casualties, and they're located at varying distances from the Macondo wellhead. One of them is, you'll see, 8 kilometers away from the wellhead, and was part of the reason why we chose it for this study. But aside from just their historic value, these shipwrecks also play a contemporary role in seafloor ecology in that they form the basis for artificial reef ecosystems by providing a solid substrate on the seafloor, something that's fairly rare in the open ocean that allows for organism settlement. And by providing this service or this habitat, they create locations of intact deep sea ecosystems with high trophic complexity. And so they create locations where we can expand our knowledge about life in the deep sea. But for the purposes of our shipwreck or our oil spill study, we also think that these are ideal locations to monitor the trophic effects of the Deepwater Horizon spill over time. And at the bottom here are 3 more of the 7 shipwrecks included in our study. These are all 19th century wooden hulled and copper-clad vessels that are also located at 3 varying distances from the wellhead. And you could see, in this case, a picture really is worth 1,000 words of how much life grows on these shipwrecks in the deep sea. And so, we are really studying in a broad sense deep sea shipwreck ecology. And I've brought in 2 of the videos from our study. This guy's real cheeky here. Up on the top is the Viosca Knoll wreck, and on the bottom is the Ewing Bank shipwreck. I was just telling Ruth about this story. You put your best field plans together. You spend a lot of time and money getting a ship and an ROV to the seafloor. You spend 2 hours looking for an artifact, and a fish decides to photobomb you. And you just kind of have to sit back and enjoy it as it's happening. But the fish does create, it illustrates my point here. What I hope that you're seeing on these shipwrecks are the visible, often charismatic biodiversity, the bivalves, the corals, the finfish. Down here with the Ewing Bank, you can see the Lophelia coral head that's growing across the entire surface of the vessel's bow. But what you can't see with your eyes, but hopefully towards the end of this presentation you can see with your imagination, is the invisible majority of microorganisms that are covering every single surface of these shipwrecks. And they provide the foundation for life in the deep sea, primarily through chemosynthesis, but in the case of these shipwrecks, they are laying down biofilms on these shipwrecks that allow other organisms to ultimately settle. So more specifically, we're studying shipwreck microbial ecology. It's a fairly new field. As far as I know, we're the only folks that are doing it, so that's definitely fun. And we're doing this because all surfaces that enter the marine environment are almost immediately colonized by microorganisms. That colonization process leads to the formation of a biofilm, and it's the biofilm of microorganisms that allow for the establishment of chemical as well as physical cues that provide for recruitment of and attachment of higher organisms. And it allows these shipwrecks to ultimately graduate into an artificial reef. But we're also studying shipwreck microbial ecology because we know that microorganisms are ubiquitous. They are numerically abundant, the most abundant organism in the ocean, metabolically diverse, and they have very short lifespans. And these principles usually mean, almost in every case, that they are the first to respond and interact with a contamination event. So we're studying these microbes because we want to address how the spill has affected the foundational communities on artificial reef ecosystems, but also so that we can address how spill interactions with shipwreck surfaces are impacting the preservation of these vessels, primarily through the lens of studying microbial corrosion processes. So shipwreck microbial ecology allows us to hit a nice trifecta in our study of time, place, and historical context. The work that supported most of the data that I'm going to share with you today was organized under a NOP, National Oceanographic Partnership Program funded study called SCHEMA. And SCHEMA stands for Shipwreck, Corrosion, Hydrocarbon Exposure, Microbiology, and Archaeology. You can't have a Gulf research program without having the bumper stickers and the acronym. And we wrestled with this acronym. It was hard to find a word that could encompass all of the disciplines that were included in this study, but mostly we picked SCHEMA because of the meaning of the word schema. It's a representative framework, and so we're using these shipwrecks as a representative framework to study spill effects over time. So the high canopy view of SCHEMA and our purpose is to document the spill's prospective lasting effects on historic shipwrecks. And we're doing this through three angles. The first one, which I'll spend almost all of my time talking about today, is how the shipwreck how the spill impacted shipwreck microbiomes through a comparative study of impacted as well as unimpacted sites. And we're primarily using sediment for this work, where we're looking at the composition and function of both bacteria and archaea, although I'll almost exclusively talk about bacteria in this presentation, just in the interest of time. But we're also coupling these analyses with sediment geology and sediment geochemistry measurements. In addition, we're also doing biofilm recruitment experiments, both in the laboratory and on the seafloor. We're also interested in studying the role of microorganisms in metal corrosion to identify any long-term effects to shipwreck preservation, also through in situ laboratory experiments and ones that are happening on the seafloor. And then lastly, the archaeology partners in this work are conducting surveys, both video and photo surveys, and also incorporating some technology by using 3D imagery so that we can retain records at the centimeter scale of certain angles of these shipwrecks so that we can monitor their deformation processes over time. I want to just talk you through some of the sites that we selected for this work and how we selected them. We used three primary criteria to select the sites for this study. The proximity to the well, which is right in you can hopefully see my cursor there, right in the middle of this map. And the depth of the sites, we were trying to normalize the site depth as best as we can, although shipwrecks, they wreck where they will, so you only have so much control on that. And then lastly, we only included in our study shipwrecks that had a pre-spill archaeological survey so that we would have some basis for before and after comparisons. And by doing this, we were able to select 7 sites, 2 off to the west at about 150 kilometers, 2 off to the east of the wellhead at 75 kilometers, and then there were these 3 that are within 12 kilometers of the wellhead. We also mined the IRMA database at the outset of the study when we were selecting our sites back in 2013 to get an understanding of the surface extent of oiling. And using this information, we were able to classify these sites in 3 areas: a heavily impacted area, a moderately impacted area to the east, and then this control area, which is outside of both the surface and subsurface plume. And I'll be referencing these colors throughout the rest of the presentation, just to keep you of mind of the different impacts that we're looking at. Since the beginning of our study, and as recent as a couple years ago, the NERDA data set, which included a community scale effort, some folks in this room certainly contributed to this, of sampling of the seafloor. The data set was released. It's been published in a few highly visible publications. And we used this data set, which included hopane, PAH, and Corexit residue data from about 12,000 cores in the Mississippi Canyon and outside of their area to look at how well we did with site selection. And what we did here was we gridded the data from the NERDA data set, placed it on a map, and also superimposed our site locations on here. And after doing this, we can now confirm that at least 2 of our sites, the Mardi Gras shipwreck, a 19th century vessel, and the German U-boat U-166, are both located in an area that has been termed the acute fallout plume, or the acute footprint. And you could read more about that description of this acute footprint in these 2 papers that I have listed at the bottom here. So for the field work, at this point we've conducted 7 cruises in service of this study, but the majority of the data that I'll present to you today were collected during 2 cruises in 2014 on the RV Pelican operated by Lumcon. And on these 2 cruises we used the Global Explorer remotely operated vehicle, seen over here, to collect all of our data. In March of 2014 we conducted video and photo surveys to document our sites. Also, we collected 3D laser scanning data so that we can look at these fine-scale imagery of the shipwrecks, and we also deployed biofilm recruitment experiments. In July of 2014, it was a sediment bonanza. It was very fun. We went and collected sediment in very close proximity to the shipwrecks, within 2 meters, and then outside of the debris field at approximately 100 meters in most cases. And we also recovered our biofilm recruitment experiments that were sitting on the seafloor for 4 months. We've had 4 as of Friday, but now as of Monday we've had 5 additional cruises in this study. Last Saturday morning, the RV Point Sur, which USM operates, was sent out into the Gulf to take a look at this oil spill that happened just last week. Since we were out there in Mississippi Canyon anyway, we decided to collect some more data to go towards the annual monitoring of these sites. And so we have quite a bit of information to track both the effects of the spill as well as the response and recovery of microorganisms at these sites using cruises on the Pelican as well as our RV Point Sur. For the work, we're primarily using sediments surrounding the shipwrecks to document the spill effects. And down on the bottom here, you can see a very sped up video of push core collection. It would be nice if it happened that fast, but it's kind of boring. When we're using sediment for this study because we know a bit about sediment dynamics and sediment deposition in the northern Gulf of Mexico. And we know in the Mississippi Canyon area through a number of publications that the annual average sedimentation rate is about 0.07 centimeters per year, less than a centimeter per year. It's a number that I want you all to keep mind of, because I'll bring that back in just a few slides. And so by knowing that value, we know that if we collect a 20 centimeter push core, as you're seeing in the bottom right-hand corner, we know that we're capturing about 300 years of seafloor history. And that allows us to capture the time frame when the shipwrecks arrived on the seafloor, whether they're 19th century or World War II era, as well as the effects of the Deepwater Horizon spill. And then we take these sediment cores and we sample them down core at 2 centimeter intervals so that we can profile geochemistry, physical properties, as well as microbial ecology. The microbial ecology is what I'll spend most of my time on in this presentation. We looked at both bacterial and archaeal, and one of my undergrads actually did some fungal work too, using a sequencing approach by way of Illumina-MiSeq sequencing. We used a custom bioinformatics program or protocol that was developed by my former postdoc to quality control the data and produce all of the information that you'll see in this presentation. We also coordinated or collaborated with Mandy Joy's lab at UGA for some of our geochemical analyses. Her lab took care of PAH and total petroleum hydrocarbon analyses, as well as radiocarbon natural abundance, so that we can look at both the fates and the effects of hydrocarbons and dispersants on the seafloor. And then lastly, we worked with the Naval Research Lab for sediment physical property analyses. The 2 that I'll talk about in this presentation are the sedimentation rates, which we derived by geochronology profiles using lead-210, and also porosity measurements. So I'll get right into the latter two pieces of data that I just mentioned, the sediment geochemistry and physical properties. There's a ton of data in this study. We have it in review right now, and I'm just going to walk you through some of the highlights in these bullets on this slide. The first thing that we learned with the study, really the first time that we set the ROV down on the seafloor near U166, is that there was visual evidence of oil snow, oil snot, oil flocculant. It goes by a lot of names, but when you see it, you really know what you're looking at. It has a gelatinous, greasy, really filmy kind of texture to it that is unlike anything that you would come to expect for marine sediment, and it stains nitrile gloves pretty nicely. And we saw that in a very thick layer at the U166 and the Annona site. You can actually see it streaking down the sides of this core on the right here. In those same intervals where we saw that oil snow material, we also observed that at U166 and Annona sites that we had anomalously high porosity. These are the porosity data down here at the bottom. On the y-axis is depth below the seafloor in centimeters, and on the x-axis is the porosity measurements. And over here is the U166, and at the surface of this core, porosity is exceeding 90%, which maybe wouldn't be so unexpected for a sample that was collected near shore, but this is at 1,500 meters, and we typically expect such a depth to have much more dewatered and compacted sediments, even at the surface. And this porosity anomaly dropped down to about 6 centimeters in this core. In the surface of both mica and U166, these are both samples that were collected in our heavily impacted area. We saw evidence of long-chain N-alkanes and total petroleum hydrocarbon residues. But perhaps the most compelling piece of data in this study came from the physical property analysis, where we conducted the lead-210 geochronology profiles. And these profiles revealed to us what can only be termed an exceptional sedimentation event at the U166. That number that I had you remember just a slide ago of 0.07 centimeters per year is what we expect. And the sedimentation rate at this site, at least in 2014, was almost 10 times that. And this was also coincident with a radiocarbon age profile that you could see down at the bottom here that showed no change in radiocarbon age from surface to the bottom of the core. And that can only happen is if you have older material being dropped on top of fresher river-derived material, perhaps older material like a fossil carbon, or if you have lateral transport of material across the seafloor. So this data set gave us some pretty definitive physical as well as chemical evidence of an oiling event, specifically at the U166 site. Now I'm going to switch gears to the bacterial community composition data. I just want to orient you to these graphs. There's a ton of information on here. On our x-axis is the percent sequence abundance of the top 50 phylotypes in our bacterial data set. On the y-axis is depth below the seafloor down to 7 centimeters. And the top two graphics are the Halo and the Ewing Bank sites. These are our control sites. The Annona and Viosca Knoll are our moderately impacted sites, and U166 and Mica are our heavily impacted sites. Mardi Gras was left out of the party here just for aesthetics. It's very hard to fit 7 of these graphics on a slide and still make some meaningful out of it, but I'll talk you through the data that we observed at Mardi Gras as well. But I'll first start with the control sites. We found at these control sites that we had the highest biodiversity and surface samples at the control sites overall. You can really see this at Halo, where we have this nice even distribution of the top 50 phylotypes across the surface of this sample. At the control sites, we also observed that there was higher biodiversity in very close proximity to the shipwrecks at about 2 meters away versus the outside of the debris field sampling. And it was that little piece of data that tipped us off that there might be some more basic research to do at these these shipwreck sites, and that's what led us down the stowaway concept, which I'll come back to towards the end of this presentation. At our heavily and moderately impacted sites, in general, we saw a reduction in biodiversity in surface sediments, and we also saw a concentration of a single species associated with the Pisciricetiaceae family. This family includes the Cycloclasticus genus of obligate PAH degraders. Up to 16% of sequences in surface layers at U166, Mardi Gras, and Viosca Knoll were affiliated with this one phylotype, and we didn't see a decline in this phylotype through those upper 6 centimeters, where we also had a porosity anomaly, where we also had that exceptional sedimentation event. And then lastly, at the U166 and Mardi Gras sites, these two heavily impacted locations, which were in the vicinity of that acute fallout plume, we observed that biodiversity right next to the shipwrecks was no different than it was away from the shipwrecks. So that gives us an idea perhaps of the aerial extent of the spill on the seafloor around these sites. So these information provide us with some biological evidence of an oiling event specifically at the U166 location. Aside from looking at just the presence and abundance of different microorganisms at these sites, we're also very interested in learning about what features are driving bacterial community composition. And one way of doing this is to take our sequence table, our OTU table, operational taxonomic unit table, and derive Bray-Curtis similarity data from that data set. And then we could take the Bray-Curtis similarity data, project it into a two-dimensional state using non-metric multidimensional scaling analysis, and this tells us how similar different samples are to each other. So if two samples or two dots on this plot are very close to each other, that means they're highly similar to each other. If they're on opposite ends of the plot, they are very different from each other. And so what I did here was coded all of the samples that were taken at those 2 centimeter intervals down core according to the site that they were collected from, and here are all 7 of the sites that we analyzed in this work. And the first question that we asked was, is there a shipwreck-specific microbiome? Does the presence of a shipwreck on the seafloor shape what microorganisms are present? And the answer to that was actually yes. You can see, especially for our 2 control sites, the Halo site and the Ewing Bank site, that they form discrete clusters that are largely containing only the samples from those 2 locations. We also see a big group of samples up here that include most of the U166 sites, and when we do the analysis of similarity on this, we find that the presence of a shipwreck is driving microbial community composition on the seafloor. So that was kind of neat to find that out. But the real thrust of this work has been how has the spill affected these microbial communities on the seafloor. And so instead, on this slide, I coded the sites according to the impact type that they had. If they were in a reference or in a control location, heavily impacted or moderately impacted. And when we do this, you can once again see that our control sites form these outgroups. But we have up here is a mash of samples that were collected from our heavily impacted sites, the Mica, the Mardi Gras, and the U166, and also a few samples from the Annona shipwreck site, where we also saw some petroleum hydrocarbon residues. And when we do the analysis of similarity on these data, we also see that the impact type is a driving factor on microbial community composition. And when we do the pairwise analysis of similarity, we find that the control sites are significantly different from both the moderately and heavily impacted sites, but that the moderately and heavily impacted sites are not statistically different from each other. So most of these data were collected in 2014, and this gives us an indication that even 4 years past the spill, we're still seeing a signature of the spill on these seafloor microbiomes. We also investigated how the spill potentially impacts community function. We did this using an in silico approach, using an algorithm called PyCrust, Phylogenetic Investigations of Communities by Unreconstructed blah, blah, blah. It's a very long title to make a very cute acronym. This is not our acronym, it's one of our colleagues. But this algorithm allows you to predict functional capabilities of microbiomes by using 16S data and referencing those data to a reference genome data set. It's only restricted to fully characterized and identified microbes, so any microbes that are uncultivated or unknown don't get included in this analysis. But even with that limited capability, we found that the abundance of PAH degradation genes was elevated at both of our heavily and moderately impacted sites compared to our control. We also found that sulfur metabolism genes were significantly elevated at those two locations relative to our control. Now, sulfur metabolism might seem like it comes out of nowhere. It's not really directly related to the oil spill, but we know that there tends to be a good amount of sulfur in crude oil, and it leads us right to the second area of our study of how the preservation of historic shipwrecks might have been impacted by the oil spill. We know one of our collaborators is one of the experts on microbially mediated corrosion. We know that microorganisms contribute to corrosion processes in the marine environment. And we also know that sulfate reduction occurs on the seafloor. It's one of the dominant pathways in sediment diagenesis, and the metabolites of sulfate reduction are able to chemically attack metal surfaces. So when we saw that sulfur metabolism peak, that really did pique our interest as well. And so my former postdoc, Jen Salerno, conducted a series of experiments in the lab to understand how crude oil, dispersed crude oil, and dispersant influence biofilm recruitment, biodiversity, as well as corrosion processes. I don't have enough time to go into these experiments. They involved metal settling plates, replicates that were incubated over a 4-month period that she sacrificed at 2-week intervals. And the big takeaway from this study is that we found that Corexit, the dispersant that was used during the oil spill, resulted in a sustained reduction in biodiversity throughout the entire course of the experiment when it was applied at environmentally relevant concentrations. But more to the point of the sulfur metabolism question, we found that the addition of oil at concentrations that were likely expected on the seafloor during the spill resulted in an increase in sulfur metabolism genes as well as phylotypes, and a significant increase in metal loss on these metal settling plates. And this is the data down here. The red line here is us tracking metal loss over the 16-week period, and you could see that significant increase in the metal treatments. We think that this is a pretty important question to address, since it potentially calls into question the physical integrity of these artificial reefs. So one of my students over at USM, Rachel Muggy, is continuing this work with water that she collected from the Annona shipwreck. Just one more thing to say about these biofilm recruitment, or the biofilms on these shipwrecks. We did a similar experiment to this lab experiment that I just described to you on the seafloor. And during our 2014 operations, we took these biofilm recruitment platforms that have replicate carbon steel discs poked into the side of them and stuck them on the seafloor within 2 meters of all of our shipwrecks. And we left them there for 4 months and then went back and recovered them and looked at the composition of bacterial and archaeal microbiomes on these surfaces. And what we found when we recovered these was something that I didn't really expect, although if I stepped back and thought about it, I should have perhaps. What we found was that the composition of the microbiomes was very distinct relative to the shipwreck that we placed the experiment next to, but we also found that there was higher similarity in community composition based on if there was a wooden shipwreck present or if there was a metal shipwreck present. So the presence of the shipwreck in the environment is not only shaping the sedimentary communities, but it's also potentially shaping the dispersal of microbiome or microbes through the water column as they're potentially sloughing off of the shipwreck. And when we looked closer at these communities, we found that the communities were distinct, and in particular, one thing popped out at us, At our metal shipwreck sites, we found a significant increase in the iron oxidizing zeta proteobacteria class near our metal shipwreck sites. They were very prominent at these locations. And so this is kind of my segue into where the work is going. What this study has told us so far is a good amount of information about how the spill has affected these shipwreck sites, but we also are starting to understand how shipwrecks on the seafloor are shaping the seafloor biodiversity, because we now have two pieces of evidence that shipwrecks might be influencing the dispersal and the biogeography of microbes on the seafloor. And so I just want to spend the rest of my time talking about this stowaway idea and where the work is going. Shipwrecks, dispersal, and microbial biogeography, that's where we're trying to take this. We know a little bit about biogeography, specifically about microbial biogeography. There was a seminal paper by Martini and colleagues in 2006 that talked about what features shape microbial biogeography. It's an understudied field because we've only in the last decade or so had the molecular techniques where we can really address questions of microbial biogeography. And we know that biogeography of microorganisms is shaped by historic events, including distance from land, isolation from other ecosystems, and also passive dispersal. But we also know that contemporary conditions, including local geochemistry, are shaping what the distribution across space and time of microorganisms. We do know a little bit about the things that shape marine sediment microbial biogeography. We know that one of the historical constraints on biogeography are distance from land, because the distance from land controls the sedimentation rate, which ultimately controls the physical properties of marine sediment in terms of grain size and porosity. And these things in turn impact how many cells are present and how many phylotypes are able to be deposited into marine sediment. But there are contemporary features on the seafloor that are also shaping microbial biogeography, including seeps, vents, wood and whale falls, and of course shipwrecks, since I've been talking so much about them. And we know that these contemporary features create environments where they have high biodiversity and high trophic complexity, the establishment of in some cases of strong redox gradients, and these things can affect habitat filtering through these environments by producing these specialized habitats, which can in turn become specialized biogeographic provinces. But I think shipwrecks differ from these other natural features on the seafloor because they're adding unnatural attributes, physical attributes to the seafloor, including in some cases a debris field, impact craters, which can later become sediment traps, and then also high relief that could potentially increase the connectivity between the overlying ocean and the seafloor. So I just want to engage you all in a little bit of a thought exercise, and I'm going to use the U166, which has been the star of our story so far, to illustrate some of these concepts. The U166, just to tell you about her history, she was dispatched to the Gulf of Mexico in 1942 for the purpose of mayhem, mostly to disrupt trade routes. She was sank only months after being deployed to the Gulf when she was depth charged by a Navy patrol vessel that was escorting a ship that the U-166 had just moments before torpedoed and sank. And when the U-166 ultimately shipwrecked, she was diving to escape the Navy patrol vessel and was moving at a rate of about 30 knots. This is what our marine archaeologists have told us. The two archaeologists that discovered the U-166 are on our study team. And this was about the speed that they estimate that she encountered the seafloor at 1,500 meters. And when she encountered the seafloor, she did so in two pieces. Over here on the right is our site map, and you could see the stern section, which is the main section of the wreckage, is broken off from the bow, which is about 150 meters to the northwest. And in between these two places is an extensive debris field that contains all manner of material, including unexploded ordnance, unfortunately human remains. This is a war grave, as well as a lot of metal and wood. And so aside from that debris field, when she encountered the seafloor, she also created a 3-meter impact crater, which she's currently sitting in right now. And when I saw this image, this 3D image of the U-166, I was almost instantly captivated by it. And I was captivated more than just by the shape of the shipwreck, but by what was missing from around the shipwreck, all of that sediment that would have filled in that 3-meter impact crater. And when I saw this image, it brought to mind something that I had seen in a basic biology class, where you have a puffball mushroom and the slow motion video of a raindrop hitting the top of the puffball mushroom, exploding it, and then suddenly all of the spores get exploded out of the mushroom, and then they're carried away on active air currents so that it increases the distribution range of that puffball mushroom. So I looked at this impact crater and I wondered, is the shipwreck the puffball mushroom on the seafloor? Did all of that sediment get ejected from the seafloor, thrown up into the water column, and with it all of the microbes that were associated with it? And this gave birth to a lot of biogeography questions about how far are these microbes transported? How long does the influence of that explosive transport event last? And really a very basic question of, do these shipwrecks form islands of biodiversity for microorganisms much the same way that we know that they do for macroorganisms? This wouldn't be very interesting if this was just an isolated case, but of course the U-166 is not an isolated case. This is the BOEM shipwreck database for the northern Gulf of Mexico. This is just the Gulf of Mexico, so put your mind around what the entire continental shelf might look like. We know that there are conservatively 2,000 historic shipwrecks in the northern Gulf. These discoveries are typically made during seafloor surveys for the oil and gas industry, which explains their close distribution to the oil and gas infrastructure. This does not limit where shipwrecks are. We anticipate that there are more shipwrecks further off the shelf, and in fact new discoveries are made every year as oil and gas energy exploration continues to move further out into the shelf. And these shipwrecks range in size, range in age, hull material, the velocity that they potentially hit the floor, and their degree of isolation from land and from each other. And for me, this is a really great setup for doing experiments on studying microbial biogeography. So the experiments that I want to do with this work are asking the questions, do shipwrecks shape seafloor biogeography? And I would like to focus that question through the lens of the island theory of biogeography posited by MacArthur and Wilson in 1967. And this island theory dictates that species richness and biodiversity decrease as a function of distance from that island-like ecosystem. So with taking our shipwrecks, we want to ask the question, is there an island effect created by these shipwrecks for seafloor microorganisms? And our null hypothesis for this work is that the biodiversity next to a shipwreck is no different than it is in the open seafloor. And the alternative hypothesis to this is that we will see a decrease in microbiome biodiversity as well as species richness as a function of distance from these shipwrecks. So we've started this work. We've picked two sites to begin this study, the Ewing Bank shipwreck and the steam tanker Halo. Both of these are in our control area. That's really the obvious reason why they were selected for this part of the work. Just to give you a little more info on them, the Ewing Bank is sitting in about 600 meters of water. She's fairly compact at 40 meters long, and this is a wooden-hulled, copper-clad vessel from the 19th century. The steam tanker Halo is in a much shallower environment at 140 meters. She's a gigantic hulk of steel on the seafloor at 270 meters long, with quite a bit of high relief. There are points of this wreck that stick up into the overlying water column at 15 meters, which is like heart-thumping if you're sticking gear around one of these shipwrecks. And what we did with these sites is we collected sediments along east-west transects at 2, 25, 50, 75, and 100-meter intervals. The 2-meter sampling was collected with an ROV, and the ones that are further out were collected with an instrumented multicore. On one of our cruises, we had a down-facing camera on it, and on all of our cruises, we've had a USBL transponder so that we can know precisely where we're placing this multicore on the seafloor. And so I just want to just share with you some of the early results from this study, starting with the Ewing Bank shipwreck. This is bacterial community composition at Ewing Bank. This is another NMDS plot like the ones that I showed you just a few slides ago. When samples are close together, they're highly similar. When they're far apart, they're dissimilar. And these are color-coded according to the distance that they were collected from the primary wreck site out to 100 meters. These little numbers that you see on top of all of the samples, these are the sediment depth below the seafloor that the samples were collected at. And so the question is, do these shipwrecks create islands of biodiversity? And is there a distinct microbiome, bacterial at least, of these shipwrecks? And the answer to that is yes. You can see in this green area, the 2-meter sampling, we see a distinct cluster of samples. We also see the surface samples from that near shipwreck site forming 2 distinct clusters. But these 3 other clusters of samples, they're not arranged according to distance from the shipwreck. If you look closely at this, you'll see that they are highly organized by sediment depth. So community composition is being dictated by sediment depth, which makes a lot of sense because this is likely indicative of redox zonation, which we predictably expect in marine sediments. And the analysis of similarity explains that the shipwreck microbiome is statistically distinct from the other distances that we collected. But the real question is Ewing Bank an island of biodiversity for shipwreck or for seafloor microbes? And the answer to this is yes, where we see richness, species richness as well as diversity for both bacteria and archaea decreases as a linear function of distance from each of these shipwrecks. So this was very gratifying and kind of cool. Switch gears to HALO now. Same display of data, bacterial community similarity. You'll notice that there's no pink group in here, the 25-meter sampling, mostly because I'm a chicken. I am very averse to sticking something blind in the ocean where I know that there's a davit at 15 meters sticking out. So we backed away from that shipwreck to exactly 50 meters. But when we did this, we still see that there is a distinct microbiome associated with the HALO shipwreck. We do see some scatter of some of those near shipwreck samples, though, in this group over here. But generally speaking, the off-site collections are, again, highly organized according to sediment depth and redox zonation. And the analysis of similarity confirms that there is a distinct shipwreck-associated microbiome. We asked the question again, is HALO an island of biodiversity? And the answer for this is kinda. In terms of archaea, we see that species richness and diversity do decrease as a function of distance, but this is not the case for bacteria. So that has us scratching our heads, and I don't really have an answer for why that's the case, but I have a hypothesis perhaps, and it comes back to this active dispersal event, this puffball mushroom, and the age of these shipwrecks. And what I did here was I created a contour plot of Shannon diversity, which is arranged according to sediment depth below the seafloor and distance away from the shipwreck. And these warm colors indicate high biodiversity, the cool colors are low biodiversity. And we see at both Ewing Bank and HALO this bullseye, this hot spot of biodiversity right next to the shipwreck, but in Ewing Bank you'll see that it's pushed down a little bit further in the sediment, whereas at HALO it's up right at the surface of the sediment column. And I'm wondering if we're seeing this because the Ewing Bank is approximately 150 years older than the HALO, and that we're still seeing this surface effect, this recent redeposition effect happening at the HALO. I'm also wondering where we see this hot spot of biodiversity out at 75 meters from the HALO shipwreck. I'm wondering if we're still seeing that flinging event of the sediment going out into the surrounding environment. This is something that I can't answer right now. It certainly requires additional study. We have 2 proposals in review to go out there and address this work, where we're going to conduct radial transects around these shipwrecks to capture the 360 dynamics, as well as capture some information about deep sea currents. And that's kind of where the work is going from this point forward. And so I just want to leave you with some thoughts about what microbial stowaways can teach us about the seafloor. I think that this is more than just a niche topic, an esoteric thing for microbial ecologists to have fun with, great conversations at cocktail parties. I think it can also tell us about how these shipwrecks are really influencing the seafloor in a more broad sense. It seems like we might have a pretty nice indicator right here of how much benthic habitat is actually afforded by the shipwreck, maybe have us think more than just about the artificial reef itself and more about how it's stretching out across the seafloor. And perhaps knowing that information and how far this microbial signature extends from a shipwreck perhaps will give us a better basis for valuing these ecosystems in the deep, dark, remote ocean, perhaps even allow us to give them more protection. But I use that word remote, and the big takeaway for me for this is I think I need to rethink what we think of as remote. These environments aren't really remote. You've seen the map now. It's the map that you can't unsee. We know that the built environment is out in the deep ocean in a big way, in places that we don't even know about yet. And so it has me really rethinking how I do my science in these remote places. So I'll leave you with that thought. And I just want to thank the many folks that have supported this work. Our primary funding came from the Bureau of Ocean Energy Management and the Bureau of Safety and Environmental Enforcement, as well as the Naval Research Lab Platform Support Program. We've received tremendous support from both LumCon as well as the Point Sur crew. We've had many collaborators, my students past and present. I thank all of them, and I, of course, thank all of you for your time and attention.